Hello, and welcome to a new edition of H.E.L Explains with your host, Dr. Mario Torres Rodrigo, and today we're going to be talking about the history of bioreactors. But the first thing we need to ask ourselves is, what are bioreactors? Well, bioreactors are vessels designed and produced to provide an effective environment for enzymes or whole cells to transform substrates into products. And here at H.E.L we have our own line of bioreactors that we call Bioexplorers. And here on this slide you can see some of them. You have a Bioexplorer 100, 400 and 5000. But we understand at H.E.L that we were not the first ones by any stretch to create a bioreactor. And actually, recent studies have shown that the first bioreactors date from 10,000 years ago in China. And these first bioreactors look something like this. Just a humble jar, clay jar. Chemical studies of the organics trapped in the clay of this jar reveal fermentation products for substrates such as honey, rice and grape juice. And even nowadays, 10,000 years later, we still ferment the same things. From honey we can produce mead, from rice we produce sake, and from grape juice we produce wine. And I've been talking about fermentation, and you might also be familiar with the word respiration, and both are biological processes that have the same objective, producing energy from organic molecules. But they have some differences. So in the case of respiration, if we have a molecule of glucose, like the one you can see in this slide, we break it down completely to generate CO2 and water. And a lot of energy is released in this process, and the cell has the ability to store the energy from these reactions into an organic molecule called adenosine triphosphate, or ATP. And the yield of this reaction is between 30 and 32 ATP. On the other hand, if we have a fermentation process, what happens is that the molecule of glucose doesn't break down all the way down to CO2 and water, and instead is going to generate another organic, such as, for example, ethanol and CO2. And the yield of this reaction, it is much lower, it's only 2 ATP. So you might be thinking, what is the point of then having a fermentation if the yield is so low? Well, the main difference is that for respiration, we require external agents, such as oxygen. So in the case of not having an atmosphere with oxygen in it, then fermentation is going to be a more effective reaction. But fermentation and humanity have a very intertwined kind of story. And I described the first evidence of fermentation in China in 7000 before Christ. But for example, in Iran, 5500 before Christ, it was the first evidence of grape fermentation of wine. Egypt, some years later, in 3000 before Christ, it is the first evidence of leavened bread, so bread that has been fermented using yeast in the dough. Tigris Valley, 2000 before Christ, it is the first evidence of pickled vegetables. In this case was pickled cucumber. And then the last example I want to highlight here is again China in 500 before Christ, in which they were the first ones to use fermented tofu kernels that had some mold in it. And what they discovered is that if you apply the moldy tofu kernels on wounds, these wounds didn't infect and they healed much better. What they didn't know at that time is that actually they were using penicillin for the first time. And it was until more than 2000 years later, in 1928, when Dr. Ian Fleming, who at that time was working in St. Mary's Hospital in London, discovered penicillin, which is the molecule you can see in this slide. And this was quite a serendipitous discovery. And what happened is Ian Fleming went on holiday, and just before going on holiday, he inoculated some petri dishes with pathogens. On the way back from the holiday, what he found is in one of those plates there was a massive blob that didn't correspond to the pathogen colonies. Though the pathogen colonies, they are bacterial colonies, and they are much smaller and they are dotted around the plate. But if we have a look at this petri dish, we find that the colonies around the fungal colony are much smaller in size. And this is because the fungus, in this case Penicillium rubens, was producing penicillin, an antibiotic that was preventing them from growing. Funnily enough, when Dr. Ian Fleming published his work, it got oversight by the scientific community. They didn't deem it that important. And it wasn't until some years later in which Dr. Ian Fleming published again some more studies, and what they did was using penicillin to treat mice that have been inoculated with a pathogen, resulting in a massive survival increase rate on the mice, that the scientific community then paid massive attention to penicillin and antibiotics. And then it became some kind of race to try to produce as much penicillin as possible. And at that time in Oxford, there were 6 ladies that nowadays are known as the penicillin ladies, that were the ones in charge of producing the broth and growing Penicillium rubens. And at this time, it was an incredibly time-consuming, resource-consuming and arduous task. It involved producing massive amounts of the broth and using whatever container you could find lying around to try to grow the fungus. This involved using things such as bedpans and even bathtubs. So I was talking about bioreactors, and I'm talking about penicillin. What is the link between these two concepts? So we need to go back in time some years to 1915, in which Hein Weizmann, who at that time was a researcher in Manchester, was the first one to design large vessels to allow microbial growth to produce acetone. And acetone at this time was very important. We are talking about 1915, First World War, and acetone was used for the production of explosives. This discovery was so important that the Prime Minister at that time contacted Hein Weizmann and asked him what he wanted as a reward for his scientific endeavors. And he asked for help for the people. He was Jewish, so he wanted the Jewish people to have their own land. This resulted in some years later the British Prime Minister signing a treaty in which he promised Jewish people to help them find a land for themselves. So we can see that actually bioreactors have also a very important part in history, so important that Hein Weizmann ended up being the first Prime Minister of Israel. Let's move forward some years to 1929, in which Harden and Young were the first ones to develop aerobic fermentations. And if you recall, when I was describing and defining what a fermentation is, I described it as an anaerobic process, a process that doesn't require oxygen. However, because biological reactions are so important, basically anything that involves using biological agents, such as whole cells, microbial cells, or enzymes, nowadays is called fermentation. So this aerobic fermentation processes that Harden and Young have been working is just after the discovery of penicillin. Paul Egley in 1932 was the first one to design stir-tank bioreactors, and stir-tank bioreactors are vessels that contain an impeller inside that is going to favor a better mixing of the fluid inside of the bioreactor, and this enhances the gas distribution inside of the reactor, but also the nutrient distribution. A couple of years later, in 1934, Strach and Schmidt were the first ones to introduce tubing inside of bioreactors with 2 objectives. First of all, using steam, and using steam you can very thoroughly clean the bioreactors, resulting in aseptic conditions, which is going to reduce any kind of risk of contamination, but also introducing air and bubbling air through the medium, through the fluid inside of the bioreactor, is going to be very beneficial to increase the distribution of gas. And finally, 10 years later, the Bees and Liebman just basically compiled all these advances that we've had since Hein Weizmann to generate the first large-scale reactor. And we're talking about 20-liter reactors. And they started producing them in mass. And what happened with massive production of these vessels is that for the first time in history, the year later, in 1945, it was the first time that the demand for antibiotics was met, and this was 7 trillion units of penicillin. So since then, there's been also more advances, and nowadays current bioreactors can be used for a huge, wide variety of applications. So until the 1950s, because of how easy they are to grow, bacterial and fungal cells reigned supreme in bioreactors. But since then, and with all the advances we have nowadays, we can grow different types of cells, such as mammalian cells. And we humans are mammals ourselves. And what happens is when we're producing certain products, such as proteins, inside of cells, they are going to have modifications on the structure that are going to depend on the organism that is producing the protein. If we have mammalian cells that are more similar to us, these modifications on the protein structure are going to be more similar to ours. And then when we use these proteins as therapeutical agents, we are going to have a reduced risk of things such as allergic reactions or rejection. Insect cells, the main advantage that they have is that they grow very quickly. So we can produce large amounts of biomass using insect cells. And algae, they are photosynthetic organisms. They can use solar energy. And what happens with this is that they can fix CO2 and generate organic molecules, such as biofuels. And nowadays, algae have a very important role in biofuels. So why is this so important? And why do we produce bioreactors at H.E.L? Well, bioreactors have so many advances, such as, for example, automatization. This means that we don't have to be paying attention and extracting things from the bioreactors all the time, but instead we have a computer linked to them that is going to be controlling the parameters inside of the bioreactor, and they are going to keep those values within the values that are needed for the cells to grow. We can increase the mixing inside of the bioreactor using things such as impellers or bubbling, as we said on the previous slide. Optimization of parameters, so we can very finely tune the conditions inside of the bioreactors, so we create the ideal conditions for the cells to grow. And finally, we can also have parallel reactors, so we have different reactors running at the same time, and because they are all individual units, then they're going to be generating, each one of them, biomass and the products that we need. So bioreactors nowadays have so many applications, and I can't talk about all of them, so I'm just going to highlight some of them here. Things such as biofuel production, biopharmaceutical production, enzyme production, water treatment, bioremediation, and one only needs to go into the literature to find that actually some of our bioexplorers have been using some of these applications, especially things such as biofuels and bioremediation. And the messages I want you to take home after this video is that bioreactors have been with humans for a very long time, 10,000 years, and during this time we have been continuously improving them. And thus has resulted in nowadays bioreactors being able to produce a vast amount of products. And that's it from me. I am Dr. Mario Torres Rodrigo, the Applications Leader at H.E.L, and please, if you have any questions, if you have any comments, just reach out and talk to us. Thank you very much.